Hi there everyone, welcome back to the channel. I'm the GCSE science teacher. In today's video, we're going to be learning some GCSE biology, and this is specifically focusing on genetic inheritance and also Punnett squares, how to analyze them, how to draw them, interpret them, all of that wonderful stuff that I hope you guys do find useful in today's video. Feel free to let me know in the comments section if this is a topic you enjoy. I absolutely love this topic. I love to teach it in real life, and hopefully you guys find this video helpful and enjoy it as much as I do. Feel free to also like this video, share it with someone else so you can share the science you love with other people. And if you'd like to join this wonderful community, please do subscribe. And thank you so much to everyone who already has. I really, really do appreciate it. Okay, so let's get started. Let's talk about genetic inheritance and Punnett squares. First of all, starting off with a bit of a recap, talking about DNA. Remember, DNA is a polymer. It's made of repeating units called monomers, and we can refer to these as nucleotides. Now, those nucleotides contain bases, A, C, T, and G. We also have the phosphate group and the deoxyribose sugar or the pentose sugar. And depending on the sequence of those bases will depend on the proteins that are made. And remember, DNA is a double helix shape. It is composed of, like I say, those bases, but it's composed of sections of genes that, like I say, code for proteins. We also know that DNA is wrapped up tightly into structures called chromosomes, which help to organize this information, because the human genome, for example, is absolutely massive. Other organisms, of course, have their own genetics as well, their own genome. And where is this DNA found? Like I say, in the chromosomes, then it's found in the nucleus, and then inside a cell. This is for eukaryotic cells, like plants and animal cells. However, there are some organisms like bacteria that are prokaryotic that do not have chromosomes, that do not have a nucleus as well. So that's just worth mentioning as a bit of a recap. Okay, so other genetic terms that I want to add into the mix now are a couple of different ones that you see here. So the first one is this idea of a genotype. Now, this is referring to the genes that we're looking at, the specific genetic inherited alleles, the information that's passed on from parent to child. Now, I've used another word called alleles. And an allele is just a version of a gene. It's a flavor of a gene. And this is inherited by our parents. So our two biological parents, our mum and our dad, biologically speaking, we're talking about here, they will essentially pass on two alleles to us. And those alleles will help us determine our genes and our physical features, our phenotype, which links to the next term here. So the phenotype is the physical features, the physical characteristics that we can see, those observable traits that are expressed from the genome, from the genotype. So our genes will determine this. Of course, however, like anything, there's always exceptions, because let's say, for example, hair color, right? The natural phenotype of someone like myself is kind of, a brunette brown hair. However, if I dyed my hair, my phenotype would change to blonde, for example. However, we're talking about the natural state of individuals, and we often in GCSE biology will talk about plants, we'll talk about other animals as well. Those are the two things. We don't often refer to humans. Sometimes we do if we're talking about genetic disorders, genetic diseases, and family trees. That's something we can explore in another video. But often the questions that they ask you about inheritance with Punnett squares are often, for some reason, talking about animals and plants. Speaking of Punnett squares, there's a couple of things you need to be aware of. So I know I've said about the word allele. We can have dominant alleles and recessive alleles. That just means that an allele that has been inherited that's dominant will override the recessive one. So it's more likely and will be expressed in the phenotype if you have a dominant allele present. A recessive allele will only be expressed if two recessive alleles have been inherited. So this therefore means recessive alleles are a bit more rarer, because you obviously need two alleles for that. Now, with genetics, sometimes there are situations where three alleles are inherited for certain traits, but for the sake of GCSE, we really just keep it to two alleles, one from mum, one from dad. And then there's other ideas. There's lots of terms here, as you can imagine. Last couple, we have the idea of a heterozygous and a homozygous individual. So what does this mean? So heterozygous, you can see we have two letters. We've got a capital H and a lowercase h. The capital H and the lowercase h represent the two alleles. So you can see here we've got two alleles, one from mum, one from dad, and they've been inherited. Now, because one of them is capitalized, that represents a dominant allele, and the other one that's lowercase is representing a recessive allele. So if you've got one of each, we call this a heterozygous genotype. However, if you've got two dominant alleles, we call this a homozygous dominant individual. Homozygous meaning two of the same. And of course, we can have homozygous recessive. So we have two of the same, but they're both lowercase, they're both recessive. And like I said before, a recessive trait, such as the flower you can see here, the petals are red, you would need both alleles for that phenotype to change. So as you can see in the other two cases, because it has the dominant allele, the flower remains white in its color of petal. However, when you have a situation where there's two homozygous recessive alleles present and they're inherited, you actually change the phenotype in this case. So that's the sort of thing we're going to be referring to today. So here is a science story for you, and we've got to go back to the mid-19th century to an individual called Gregor Mendel. He was a monk who is a very religious individual, spends a lot of time dedicating their life essentially to God and religion. And he was very interested in growing plants, specifically pea plants, in his spare time as well, in the grounds of the church that he was a part of. And he noticed a few different things. So he had some red flowered pea plants and he wanted to pollinate them with white flowered pea plants. And you'd think, if you think about color theory, if you mix red paint with white paint, you'd make a kind of pink color. However, he actually found that when the red flowers were pollinated with the white flowers, all of the offspring plants were red. Now, this made him think about why that happened. And what he did was he actually bred all of the offspring plants together. And he found that most of the plants that were in the next generation were red, but some of them were also white. And that really made him start to think about how different characteristics are passed down from one generation to another. And then he came up with this idea of dominant alleles and recessive ones. So in this data, you can see that the red plants are the dominant phenotype, the dominant characteristic, and therefore the dominant allele. And the white flowers are the recessive one because, of course, we know now that two alleles are needed for white flowers, if it's recessive, for example, to be physically shown in the phenotype. So this is the sort of thing that he was able to deduce from his simple experiments that he did in his spare time, which is really cool to think about. So with all this being said, this idea of cross-pollinating red flowers with white flowers. This is what we call a monohybrid cross, where we have a specific trait that we're crossing over, genetically speaking. We're seeing what the likelihood outcome is of individuals when they are the offspring of two parents with specific alleles. So as you can see here, we have a picture on the left-hand side of a Punnett square, and we have some information on the right-hand side. So let me talk this through to you, and I'll give you some examples as well, so you guys can have a go as well. So the first thing we've got here, like I say, we've got Punnett square. We've got on the top side, we've got the mother's alleles or the genome or the genotype. We've got lowercase h, capital H. And on the left side, on the side, it says father alleles, and that's just the genotype of the father. So as you can see, the information we have here, black fur in a sheep is a dominant trait given the symbol capital H. White fur is recessive, lowercase h. I just want to point out, by the way, letters like H or any other letter in the alphabet, it really does not matter what you use. If you ever get asked to draw a Punnett square or they give you a certain letter, don't worry about the letter at all. It doesn't matter. It's just a random thing to represent dominant and recessive. What you're looking for is the capitalized letter as dominant, lowercase as recessive. That's what you need to look at, really. So anyway, you can see here that in blue, all of those four individuals, these are the possible likelihoods of the offspring having black fur or white fur as the sheep. So as you can see, if we do the cross, if you were to match these bits and pieces up, you can see that we actually have black furred individuals as 3 out of 4 chance, or 75%, and white fur as 25%. This is often the case. Recessive alleles are less commonly expressed in the phenotype, and that's the sort of thing we'd see. You would want to, in the GCSE exam, write either a percentage or a fraction, like I have, as a probability of the likelihood. Something to mention, by the way, this is just the chance, generally speaking. If the individual had another offspring, you'd have the same likelihood of chance. Just because, let's say, one sheep had a black-furred sheep offspring, it doesn't mean that the next offspring they have is going to be a white-furred offspring. It could, again, be a black-furred offspring. It doesn't matter. The egg and sperm is just a random event. So that's always going to be the case. The likelihood is going to be 75% black and 25% white fur. And this is just for this random example that I've made up for you guys. Okay, so let's do another example together. So that was just a bit of a run through with the previous one, but let's go step by step. We've got two birds. We've got one with brown feathers, which has a dominant trait, Q, and the other bird has blue feathers, which has a recessive trait, lowercase q. So we know that two lowercase q's represent a homozygous recessive individual. That means that blue feathers will be expressed because you need those two recessive alleles for the homozygous recessive traits to be expressed. We can have a heterozygous individual, like the mother. As you can see, they've got a capital Q, lowercase q. And because there's a dominant allele, that means that the mother has the brown feathers, and any offspring with that particular genotype, they will also have brown feathers as well. And other individuals that have brown feathers are, of course, the homozygous dominant individuals, so two capitalized letters or two dominant alleles that are inherited. So let's do this cross together and see what the outcomes are. So as you can see, we have filled in the Punnett square. This is the sort of thing they could ask you. They'll give you some information and they'll ask you to fill in the Punnett square and then interpret it. So this is the Punnett square that I've done. If I was to do this in the exam, I'd write in each of those alleles for each of the individuals. There's 4 potential offspring. And I can see quite easily here that we have 2 individuals out of the 4 that have the dominant allele, so they will both have brown feathers. However, the other 2 have no dominant allele, and therefore they will have the blue feathers. So it's a 50-50 chance of the offspring having brown or blue feathers, and that's the sort of thing you'd want to write either as a percentage or as a fraction, like I said. So here is the final kind of question they could ask you, especially if you are a triple student or a higher combined student. They will often give you the information and expect you to draw your own Punnett square from the information and then interpret it as well. And this can be worth anywhere from 4 to 5, even 6 marks sometimes. So it's worth knowing how to do this if you're just given the information. So let's have a look at the question. We have 2 individuals. We've got a dog with long fur, which is the dominant trait, and we've got a dog with short fur, which is the recessive trait. Now we need to remember in this case, recessive alleles, you need 2 of them for them to be expressed. That's very important. We also haven't been given a letter to represent this. So you can use any letter. I recommend using a letter that's really easy to see, which is capital, which is lowercase. One of the tips I do give students, if they give you a letter that's really rubbish, like, I don't know, S, for example, capital S, lowercase s, just make it super obvious on your page, like massive S for dominant, lower, small, teeny s for recessive. That way it's just easier for you in the exam to actually know what you're doing, because it's easily done. If you've got letters like an S, for example, or an X, it can be really hard to distinguish between dominant and recessive. So just be careful. In this situation, choose a letter that's easy. And then here we go. It says, calculate the likelihood of a dog with long fur being born when its parents both are heterozygous. Now, notice in this question, they've not given you the meaning of heterozygous. You've got to remember it. So heterozygous, hetero meaning different. You know that both those parents therefore have different alleles, so one of each. So let's do this together now. So here you can see I've just filled in the Punnett square. I've drawn this myself. Remember, they won't give you that Punnett square. I've just drawn this for the sake of the video. And I've put in the letters. We've got capital T, lowercase t. Again, a really nice letter to use because it's easy to distinguish which is the dominant, which is the recessive. Obviously, both the parents, mother and father, they are heterozygous, so that's why they've got one of each allele. And then what you want to do is you want to fill in your Punnett square. So let's do that now. Okay, so as you can see, I've just filled in my Punnett square. You can see we've got the information here. It's really important to just double check you have this correct. Lots of students will rush this. Please don't. Give yourself a bit of time just to think logically, because all you're doing is you're matching each of those together. So capital T, that makes the two capital T's together. That's all you're doing. Just give yourself a bit of time just to make sure you know what is being done. Also, at this point, I would recommend, as soon as you fill this in, you want to start to circle things. One of the things I like to share with my students is, if you notice two capitals, maybe circle it and label it. If you can see any dominants, circle them, so it makes it really easy when it comes to interpreting it. So if we have a look at the Punnett square after the interpretation, you can see that 3 out of 4 of those individuals have the dominant allele. One is homozygous dominant, and the other 2 are heterozygous. However, they all have that dominant allele, and therefore that will mean that they would all have long fur. There's only one of them that has both recessive alleles, and therefore they are homozygous recessive. That means 1 out of 4 offspring will have short fur. This is obviously used in the case of GCSEs, this idea of Punnett squares, but actually this is used for genetic counselling as well, and we can use this to predict the likelihood of individuals inheriting disorders or diseases, which is something I'm going to make a video on very soon, because this is something that you need to know for GCSEs as well. So yeah, feel free to stick around for that, because it will be a very interesting one. Links nicely to this topic too. And that's it from me today. I've been the GCSE science teacher, and you have been curious. If you did enjoy this video, please let me know by giving the video a thumbs up. It really does help me out and helps me know if you guys do like this sort of content. Feel free to also share this with someone else so they don't miss out on key revision. And also, please do subscribe. I would love it if you joined our community. Thank you so much to everyone who already has. It's amazing to have you as part of it. And if you'd like to hear more from me and you are wanting more help with GCSE science, feel free to check out my other videos. I have lots on biology, chemistry, and physics in different playlists for you. I also have broken down all of paper 1 and paper 2 content in under 30 minutes. So feel free to check those out as well. And if you'd like more short bite-sized revision, when you're scrolling through Instagram or TikTok, feel free to join me on there as well. All of it is at the GCSE Science Teacher. Thank you so much for all your support, and I'll catch you in the next one. Take care.